And usually I try not to record the announcements. And for this particular class, it's going to be best on the recording to not share the screen and just get the board notes over here. Okay. And I'm also going to leave the lights on because I think that'll make it easier to pick up the board notes. So this particular slide set, we're going to use this the whole rest of this unit. So we use about 10 slides a day or some 10 or 12 slides today, and then some next Wednesday. And then I think we have a catch-up review day, and then you have an exam the second Monday after this. So what we're going to be, the part we're going to be most on today is what is called environmental effects or environmental factors. Bacterial growth is next Wednesday, but you'll use these same slides for both topics. And before we get to environmental factors, we need to talk about food, bacterial food. All of the medically important microbes, or our bacteria studied this semester, and our parasites that we study this semester toward the end, all of them are going to have some kind of food source that we think of in the traditional way. So from a nutritional standpoint, they are similar to us. They're going to use something that is food, and that food is used for chemical building blocks. And that food is used also as an energy source. And that makes all of the bacteria that we study this semester fall into a class called chemoheterotroph. Chemoheterotrophs. Is their nutritional category. We are chemoheterotrophs. So like what do you got in the bowl? Grapes and crackers. So Gabri's grapes and crackers, when she eats them, the grapes give sugars and water, probably. And the crackers also give sugars and water, and she's going to break those down and get energy out of it, but she could also use the breakdown products to build more Gabri. All of the organisms that we talk about in this class are the same nutritionally as us. They need a food source. And they like to infect us because we already have nutrition for them to steal from us. They're chemoheterotrophs. There are also microbes that use sun for energy. And maybe they use CO2 for their carbon source, and they act almost like a plant from their nutritional needs. Those are autotrophs or phototrophs, and we don't study them in this class. Everything we're studying is considered chemoheterotroph because they need some kind of food like we do. We are their food a lot of times. So that's a little side topic about nutritional definition of microbes. The next items that we're going to talk about have to do with what are environmental factors that affect microbial growth. Can't spell this. Environmental factors that affect microbial growth. And these are the conditions. So you could call them environmental conditions. One of them is going to be what we call osmotic effects. One of them is going to be temperature. And the other big one that we'll talk about is oxygen requirements. So you can see up there it says number 3, osmotic effects. I moved it at some point from number 3 to number 1. So it's one of the big 3 that we're going to talk about. There are also pressure and pH requirements, and I don't teach those in this class, but they also are things that affect microbial growth that are in the environment that they live in. These 3, osmotic effects, temperature, and oxygen requirements, you need to know details about. And these 2, you will have some specific exam questions about those 2. And we're going to do some drawings and some kind of like problem-solving, in-depth info about those 2. So that's just kind of a preview of what's coming up. Microbes are, even if they can swim with their flagellum and they can technically go from place to place, it's really hard for them to escape something. Like if I'm a microbe and this room is too hot, I'm not going to probably make it to a place that's cooler. It's just too far. Like I'm not. So they either have to deal with the conditions and survive, or they're going to die. So let's add that onto here. They either have to tolerate the environment, or they're going to die. It's harsh. Like they can't really pick up and move to a better spot. So by changing what the environment is, we can kill a lot of microbes. We can literally make it too hot for microbial growth. We can make it too salty. We can change their oxygen conditions sometimes and kill them because they're not going to be able to get away. They won't be able to tolerate what we do to their environment. So understanding how these affect microbes lets us know how to kill them. Is one reason you learn this in this class. So the first ones of these sounds the worst. Osmotic effects, and this has to do with osmosis. So if osmosis is one of those things that set off alarms in your brain because you hated it before, I feel like I try really hard to teach those things well because I remember what that feels like. Like, oh my God, osmosis. No. Yes. Osmotic effects are really important to how microbes live. So most microbes live in a watery environment. They might live on the surface for a while, but a lot of times they're in a watery environment. And always a watery environment is going to have solutes in it. So if you're in an aqueous, watery environment, there are always 2 things present. There's water, and then there's stuff dissolved in the water that we can call solutes. Microbes also have water plus solutes in their cells. They also can be considered a watery environment. So the microbe's water and its solutes are a lot of times in some ways connected to the outside water and its solutes. Like upstairs in the toilet, I'm sure there's some microbes right now, and they're sitting in toilet water. And what happens to them in the toilet water depends on what the toilet water is like. That's the environment that they're living in. And what we want to know is, which direction does water move? Between the outside of the cell and the inside of the cell. Because that's a really big deal to microbes. Is water trying to come in and burst me? Is water leaving my cytoplasm and going to kill me that way? It depends on what this outside environment is. It depends on the direction of osmosis. It can be deadly. And we can use it on purpose to kill microbes. So this picture up here, you've probably seen that from like high school biology, or if you had Bio 1 and you did osmosis experiments. And seeing this concept of a semi-permeable membrane. Cell membrane that surrounds a microbe is semi-permeable as well, and water can go across it in either direction. If the cell has peptidoglycan around it, which a lot of bacteria do, here's our peptidoglycan. Peptidoglycan, this is already in the slide. Peptidoglycan is going to help prevent bursting of the cell. So I'm going to be using this picture a lot, together with the pictures coming up, to describe when are microbes in danger from dying from their conditions around them. We're not going to do these definitions. That's like a Bio 1 thing, so you can just draw a line through the definitions. We're going to go to this scenario. Okay, so in each one of these pictures, it's showing there's a cell, and the cell has water and solutes inside it, and the environment it's floating in has water and solutes inside it. And there's going to be an example of a cell that has a cell wall and a cell that doesn't have a cell wall. And then up at the top of the picture, it tells you the name of this kind of solution is this. So we have isotonic, hypotonic, hypertonic. And this is one of the topics that I didn't get when I was I found it really confusing when I was a student. And then I tend to teach those things slow, really slow. So we'll start. Every solution inside and outside has a % water and a % solute. Every solution in the whole world that's made out of water has some % water and some % solute. If you guess, what % solute are people cells? 30? We are 30% not water in our whole body. 70% of us is water. In the cytoplasm, it's lower than 30, how much is dissolved. And it's actually equal to the concentration of an IV bag. And if you work in medicine and you start an IV or you give people fluids.
Either 2% or 5%, right?
It's about 1%. So our cytoplasm is about 1% solute, 99% water. It kind of depends on the person's individual problem, what level of IV bag they're going to get. It's about 1% solute. That's similar to most of the bacteria that infect people. Because they live in us, they have about those conditions. So we're going to use this as our example. Okay, if this bacterium is in isotonic, like shown up there, isotonic solution, what is the percent water and percent solute in the solution it's floating in? If it's isotonic. Same, like iso means same. So we can write that in. Isotonic solution, you have equal concentrations. And water goes evenly in both directions in and out of that cell. So if the cell doesn't have a cell wall and water is going in and out the same in both directions, does this cell get bigger or smaller or stay the same? It stays the same. This cell stays the same. So does a cell with a cell wall stay the same. So that's pretty easy to understand. That's isotonic. Let's look at a different one. And we're going to change the numbers on the drawing now, at least for the solution. The second thing that you could have happen is a different kind of solution that we call hypotonic. Hypotonic. What's hypo? Below? Less than? Yeah. So hypotonic hypo means less. The tonic part means solute or the dissolved stuff. Hypotonic solution. Less solute. Less solute than what? Less solute than what the cell has. That's how the solution gets its name. Hypotonic solution. What's a possible number that could be the percent solute if this cell is in hypotonic solution? What kind of number should be here if this cell is in a hypotonic solution? Don't say it yet. Okay, now you can take a stab.
Be like 0.5.
0.5 would work. 0 would work. Anything that's less than 1% here would make this solution hypotonic compared to the cell. So make a note. I'm going to put this in a different color. The words isotonic, hypotonic, hypertonic. Those are going to describe a solution compared to the cytoplasm of the cell. Compared to the cytoplasm of the cell. So you're always comparing, well, what's this one? What's this one? That's where these are comparison kind of words. So I'm going to use 0.5 as the solute concentration in our example hypotonic solution. So if our cell has 1% solute in the cytoplasm, 0.5 is less solute out here than inside the cell. So if that's the solute concentration, what's the water amount?
99.5.
It's the rest of all of it, right? So the water amount would be 99.5% water. Once you have those numbers, then it's easy to predict where does water go? How is water going to move? Do you have a question? No. I noticed when I was going back and watching the videos that I made the last couple weeks to make sure they're okay, that I do this all the time. I didn't know I did that. Like as a, if I'm talking and thinking, I'm like, well. Okay, so if we know that we got this much water outside and this much inside, now we can know which way is the water going to move. Which way is the water going to move? From high water concentration to low water concentration. That's what osmosis is. It's water moving down its concentration gradient. So if a cell is in hypotonic solution, which way does water tend to move?
Towards outside the cell.
Towards the outside of the cell? It actually moves toward the inside of the cell. Water moves from where it's high to where it's lower. Hypotonic solution, water wants to rush in. Water wants to rush in. And that's why we got these arrows pointing in. Water wants to rush in. And if the cell doesn't have a cell wall, water will rush in and it will eventually burst that cell. There's a lab you can do this with blood cells and just kind of put them in pure water and watch them under the microscope pop open. Kind of gross, but kind of cool. Bacteria are going to be protected from that because they have a cell wall. They're not going to burst in hypotonic solution. So water will come into them and they will swell up, but they won't burst. So on this slide, if you've got the pictures and you can write on the pictures, up here, you can write the cell wall prevents bursting. And down here, cells without a cell wall are going to burst or break open in hypotonic solution. So we would not use hypotonic solution to kill bacteria. They'll just kind of swell up, but they're not going to die from that. Hypertonic, though, is a different story. Hypertonic solution, we have the opposite situation. So hyper is meaning more, tonic is meaning solute. Hypertonic. What would be a number that I could put here in this space if I say that this cell is in a hypertonic solution. It has to be bigger than 1. So the definition of hypertonic is more solution than the other one, which is the inside of the cell. So anything more than 1, and I'm going to make it 7, because I feel like it, 7% solute. So if this cell is floating in a solution that's 7% solute, then you can fill in the water concentration or the water percent. Which I bet you just did. 93% water. And then we can look, okay, if water is always going to move from high water to low water concentration, a cell in hypertonic solution is always going to shrink because water is leaving it. So if you have a human cell, a red blood cell in hypertonic solution, water will leave it and it will shrink. Bacterial cell, it's got a wall around it. The wall doesn't prevent water from leaving. So the bacterial cell will shrink. And we can change the what happens info over here. So bacterial cell, the cytoplasm inside the cell wall is going to shrink, and the cell is going to die if that persists. Same for cell without a cell wall. Cell shrinks and dies. You know how people now say that if someone is kind of mean or sassy, that you say that they're salty? That's like useful for teaching this. So you can make a solution so salty that it kills the cell that it's floating in. Like if you were in an environment, everybody around you is so salty and you just can't take it, you give up and you die from it. You can make a solution so solute, either with salt or with sugar, acid, that the cell inside it dies. And people figured this out like way long ago, before they even knew that there were microbes. That if you get a food really salty, like bacon, ham, pickles, what else is salty? Salted fish, that it doesn't go bad. Or if you put enough sugar in something like candy, jelly, honey doesn't go bad. It stays good because it's so solute that the microbes can't survive. Hypertonic preserves, because hypertonic kills microbes. That's cool. It's a way to kill microbes. Get it so salty, so solute. So here's how I kind of ask about this kind of thing on the test. We'll do an example. Okay, so usually on this kind of question, I will say something like there is a cell with a 1.8% solute concentration in the cytoplasm. It's in an environment with a 4% solute concentration. bacterial cell. I always ask about bacterial cell. What happens to the cell what kind of solution is the cell in? And then normally I like for you to do a drawing that shows the movement of water. If you really are good at this kind of thing and you just want to tell me what happens to the cell and what kind of solution is that in, if you've got that right, you don't have to do a drawing. But man, it helps me in my life all the time to do a drawing if I'm trying to figure out something. So that's what kind of question.
That would be a hypertonic solution.
It is hypertonic. Yeah. So
So the cell would shrink.
the cell would shrink and die. And you didn't have to do a drawing. So I'm like impressed. So if you had, the cell is going to shrink and die, and it's a hypertonic solution, that's really what the question's about. And then I would like discard drawing points. But if you do a drawing, that's worth some points, and get one of these right but not the other, that's good. Like you're showing me some knowledge. So to kind of quickly fill this in, okay, so the way that I do this, I fill in the water percents, and then I know that water is going to go from high to low. Water's going to leave. This solute is more than this solute, so that means this is a hypertonic solution. The cell's going to die. So you'll have one problem kind of like that on the exam. And you can make up like however many practice ones that you want. That's how those go. Osmosis is life or death for microbes because they can't get away from their solution. They either have to live with it or they're going to die. So making the environment salty is a good way to prevent microbes from growing outside the body. So that's our osmotic effects stuff.
Is the hypertonic solution, is that the only one that the cell will show up and die?
Yes. Hypertonic solution is the only one where cells are going to shrivel up and die. Yeah.
That's just the water.
Yeah, because if there's more water in than out, the plasma membrane is not going to stop water from going out. In human physiology, if you get a little bit dehydrated and your solute levels get out of whack, you lose water out of your cells too. And that causes like confusion and a lot of problems. It's the same thing. We just hope we don't die from it. And then we can realize that it's happening and drink water. Microbes they're much more simple and much more limited. Yeah.
So an isotonic solution would be like an equal amount?
Mm-hmm. Isotonic, it's the Goldilocks situation, like everything's perfect, everything's the same. And so you might have Gatorade with you. Anya has a Gatorade bottle. So I was at Food City when Moses was sick, and I didn't know that I was sick. And I was trying to buy Gatorade, and a lot of stores are out of it, which is scary. You know, why are we out of Gatorade? So many people have COVID. And I was trying to hurry, and I noticed up at the top of the, where they have the signs, like this is what's in this aisle, and it says up there, isotonic. You ever notice that on the Virginia? No. Other countries market drinks like Gatorade as isotonic because they assume that everyone knows what that means. In the US, not everybody all the time knows what that means, but drinking Gatorade is mostly close to isotonic with human body. So when you drink it, it gives you the right amount of water and the right amount of dissolved stuff in the water. Isotonic. I never put isotonic on the test. I never have. Like, I feel like it wastes your time. Like you won't have a test question where everything's isotonic because, like, I don't know. I feel like it wastes your time. So you'll either have one that's it's either going to be hypertonic or hypotonic. Good questions. If this freaks you out, like, come see me and we can practice it later. And we already did predict the movement of water. Okay. The next one is much easier to understand and not very complicated. Whatever temperature a microbe is in, it can't change the temperature. It can't run away from it fast. So it's either going to live or die or suffer at that temperature. But there are microbes that live in every temperature almost on the whole Earth. So if a microbe lives in a certain temperature, it's adapted to that temperature. Every microbe has a temperature that it prefers to live in. We say every microbe has 3 cardinal temperatures that are just part of its lifestyle. And those are optimum, minimum, and maximum. Optimum, minimum, maximum. I feel like this lecture ought to be called Goldilocks microbes or something like that. Like, is it too hot? Is it too cold? Is it too salty? Is it too watery? Or is it just right? Okay. So we're going to be looking at graphs about microbial growth. And on the y-axis, it's saying how well does the microbe grow? The taller, the better it's growing. And then on this axis, we've got increasing temperature, and this is degrees Celsius. This is degrees Celsius. And every individual microbe, it's going to have a, I can't remember what this is called mathematically. A peak? It's not a bump, but it is a bump. They're going to show that kind of graph. And the temperature at which they grow best and they grow the fastest is the optimum temperature. Optimum is temperature at which the microbe divides, grows the fastest. Optimum temperature. Minimum temperature is the lowest at which the microbe can divide. Less than that, we consider the microbe to be dying. So minimum is like, how cold can it get without dying, basically. Maximum is the opposite. What's the highest temperature at which the microbe can divide. And if you get much hotter than that, you can consider that you've killed the microbe. So we also, people like us, we have 3 cardinal temperatures. What would be our optimum?
68.
That's mine too, 68 degrees. Like the one that you feel like you live the best and is the best for you. So it's like somewhere around room temperature. How cold can people, cold. What's the lowest temperature people can tolerate? I don't actually know this. Like, I'm no like wild wilderness. I'm not a wilderness survival person, y'all.
I would guess without like a jacket, like 30 degrees.
Yeah, like around freezing. We do so much to control our environment temperature, it's hard for us to think this way. Like, I'm going to put on a coat. I'm going to turn on the heater. I'm going to jump up and down and get warm. Bacteria don't have that. How high can our fever get before we are about to die? Internal. 100 something, 100 and something, internal. So every bacterium has these 3 cardinal temperatures. Here's the whole picture of the different groups of bacteria and what their cardinal temperatures are. So we name the group that match us, like that are like people. We call those mesophiles. Meso meaning kind of in the middle. And that's the green line. So here's the temperature curve of a mesophile. And the favorite temperature of a mesophile is human body temperature, 37 degrees Celsius. So most medical important bacteria are mesophiles because they infect people, and we are their heaven, right? Our temperature is their optimum temperature. Most mesophiles, if you get them below 10 Celsius, they don't do well. How cold is a fridge in your house? I wouldn't have known this at your age either, because I didn't have a thermometer in the fridge. It's like 40 Celsius, 40 Fahrenheit. Yeah. So we keep refrigerators colder than the minimum temperature of a mesophile, so that bacteria that infect us won't grow well there. That's where these temperatures come into play. If you get mesophiles hotter than their maximum temperature, they're likely going to die. And that's kind of where our food cooking temperatures come from. Like if you're supposed to cook the chicken to 160 degrees Fahrenheit, get it so hot that most of the microbes that infect us will die. That's where the practical applications of these come in. So there's some microbes that being in this room would kill them. Is that weird? Like this group that we call psychrophiles. Psychro means cold. This room is so hot for them that they would die. I feel kind of hot, but I'm not about to die. There are microbes that live in weird places in nature that prefer it incredibly hot. They're called thermophiles. This room is so cold for them that they would die because it's not their adapted temperature. For our class, we study the world of medical microbes, and a fridge is cold enough to keep microbes from growing, and when you cook food, it's hot enough to keep microbes from growing because we're we're focused on health and medical microbes. There's a whole world of weird microbes that live in ice, or they live in boiling hot water, and that's what they like. And I find that cool, but we don't get to learn about those in here. Great. Now we come to gas requirements, and there's something that I want you to write at the beginning of this section. Which may be shocking, because I feel like when you're very young and you learn about life and living in cells, you learn that oxygen is a requirement for life, and it's a requirement for our life, for sure. But not all life forms in the whole world use oxygen or require it, and there are some microbes in the whole world that are killed by oxygen. And sometimes that's kind of hard to get used to, but it's really important in bacteria world. Do you like oxygen? Do you just tolerate it? Are you killed by it? Or do you need a Goldilocks amount of oxygen? So we treat oxygen like it is the most important thing on Earth, because for people, that's what will kill us first, is if we run out of oxygen. But there are some bad things about oxygen. Oxygen, living in an oxygen world means that you have to, oh, I'm supposed to write these down, okay. You have to deal with a class of molecules called reactive oxygen species. Reactive oxygen species, which may be sometimes abbreviated ROS. And you may have heard these called sometimes free radicals. Free radicals. So a molecule that is a free radical has an unpaired electron. If your brain just went into alarm mode at unpaired electron, not going past that in this class, but that's what a free radical is. So here's some examples. Peroxide, superoxide, there's one other, peroxide, well, I'll just write that down for right now, having a brain freeze. Those are dangerous. Those come along with living in a world that has oxygen. So there's a big so that comes from this. If an organism, including us, is going to live with oxygen, like oxygen gas in the atmosphere, that organism must have enzymes that deal with reactive oxygen species. If you don't have enzymes to help clean those up in your cell or in your body, you can be destroyed and killed by the reactive oxygen species. One of the enzymes that microbes that we study in lab have to help them with this is something called catalase. catalase. And we learn more later about how catalase actually works, but you can think of catalase as a ROS protecting, well, it's really an ROS destroying enzyme. And having something like that will let a microbe live in an oxygen environment. I need to erase this, start something else, I'm making sure. Do you have a question? Stretching your hand. Yeah. Okay. Okay, so there are 5 categories of microbes classified by their relationship to oxygen, and they're shown on this chart. This chart works a lot better in a book than it does on a slide because you can't read the words up here. But I'm going to go through the 5 different categories of microbes classified by their oxygen requirements, and we won't finish that today. But this concept is really important, and you should put a star by it in your notes about how this concept works. And I think what I want to end on is to describe what these tubes represent in this picture, because this is a little bit of a weird concept. Okay, there's a certain kind of broth tube that you can produce. I want it black. And these contain a liquid media. This is the cap up here. And in these tubes, they contain a special type of liquid media that has an oxygen gradient going from the top to the bottom. These have oxygen gradient. So up here, there would be high levels of oxygen, like the same as the air we're breathing. Down here, at the bottom of the tube, there would be no oxygen. And then as you gradually move down, the oxygen percent in this tube declines. This situation is shown in these cartoon tubes over here. The other important thing to know about these is that when you see in these tubes in the cartoon, the yellow is where where bacteria are growing. So the yellow places on these cartoons are places where bacteria are growing. Alyssa Talabat, can you turn off the light closest to me? Okay, these are real life pictures of such tubes. They still have a cap, they still have liquid media. I don't know why they have different levels. I just noticed that this year. It's kind of lame. But wherever the top level of the broth is, there's high oxygen, and at the bottom, there's low oxygen. And in real life, non-cartoon tubes, you look for where is the tube cloudy. So see how this one, it's only cloudy way up at the top. This one, it's cloudy at the bottom. This one is cloudy halfway down, and this one is cloudy all the way down. That's how they look in real life. So this kind of picture is the real life version of these cartoon tubes here. So we can see that there are, the way that the 5 categories of bacteria grow is different because they have different oxygen needs. So this one can't live without oxygen. This one doesn't do well with oxygen. This one seems fine with anything. This one looks kind of the same for right now, but it's different. This one needs Goldilocks, just right level of oxygen. That's the setup. So on Wednesday, it's a long time. Wednesday, we'll start there and we'll learn about like why do they grow that way and what do we call them? And what is their problem with oxygen? That's what we'll do Wednesday. No class Monday for Labor Day. Yes, lab on Tuesday. Yes, lecture on Wednesday.